Okay, this is from Monday, January 13th, Cell Bio.
Now, as I mentioned, that we do run a research program, so I know many students that are interested in getting research experience, and I certainly encourage them to do so. However, I do get a lot of interest in students wanting to volunteer in my lab, and as much as I'd like to take you guys, unfortunately I can't. So just due to funding issues, I simply don't have enough money to take on any more students. So I'm just going to put that out there. But I do not discourage you from approaching other labs and doing field school medicine. And it's a really great opportunity. That's how I got interested in cell biology. Just like this one. So today I'm going to talk with a little brief introduction about cell biology, somebody who's use for studying cells. Because of the snow day last week, we lost the lecture, so I'll try to get through these first two. Before we have the test on so this is and then I'll continue on with the variety of topics afterwards, and then we'll have the second test. And I just, as I mentioned at the beginning, well, after this second test, Dr. Daniels will come on and give his full 12 lectures, and I'll come back at the end and we'll go on cell survey. So today we're going to understand why do we want to study cell biology, how do we go about doing it. So cell culture is a really critical technique. And then we'll talk about the difference between normal cancer and stem cells. Okay, so the textbook, this is, I have a feeling you're going to have problems, you always do every year. These numbers are based on a physical hard copy of the textbook. Some of you may have that. Many of you probably opted for the online version, which is fine. There's been a problem with the pass with the paper and not actually done. If that happens, let me know. If it does, if this is the case, this is why I have given the subheadings here. So if you're using the online version and the numbers don't match up, you know what the subheadings are, and those are the sections that I would recommend that you read. Now, you are not tested on, there's a lot of extra information in the textbook. You're not tested on that. You're tested on what I present in the lecture. So don't freak out and say, oh my God, this is a big long chapter and I have to be responsible for it. No. The textbook is simply a resource to help me explain and clarify some of the concepts. I use the textbook to help guide these lectures. I also bring in additional information that's not covered in the textbook. So you're tested on what I present in the lecture. The same goes true for Dr. Daniel's lectures. Okay, so why do we want to study cells? So I already kind of alluded to that in the introduction that a lot of people are really interested in genes. In order to understand how a gene works, you have to understand its function within the context of the cell. The cell is basically the fundamental unit of life. And if we're interested in studying tissues, for example, in multicellular organisms, like a human, an animal, or even how organs function, we can't really get a clear idea until you go down to the cellular level. So that detailed level of understanding how cells that all collectively accumulate with a specialized function within an organ, and then understanding how the organ functions itself, you've got to step back, go down to the cellular level. My research is focused on basic research, but also disease context. One of the diseases I study is Alzheimer's disease. And trying to understand a disease, you need to actually go back to see what the non-disease state is. So looking at a cell that functions under normal circumstances is really important to understand when those normal functions don't happen. You can enter into an abnormal or disease state. So it's really important to understand the fundamental principles of how cells work. And these fundamental principles led to multiple Nobel Prizes in cell biology, and which I'll show here. Okay, so you're not going to be tested. I'm not going to memorize every single Nobel Prize. I bring this up because I would say last century was the sort of at least the last half of the 20th century was very gene-centric. Genes were DNA and mutations were really kind of the big thing that people were after. I'd say in this century, it's the century of cell biology. And this is exemplified by, and actually I need to update it, and we're a little behind, there's a couple of other ones that I haven't included here more recently, but since the 2000 and onwards, multiple Nobel Prizes in either physiology, medicine, or chemistry have fundamentals related to cell biology. And everything that's highlighted in red are topics that we will cover either directly or indirectly in this course. So that's, again, just trying to reiterate how cell biology really sort of widens research and therapeutic development in the next 25 years, and will continue to do so probably the next 25 years. So the expectation is if you're taking this course, you've taken high school biology, you've probably covered some aspects of what a cell is and eukaryotic cell. You've taken first year university biology, which would cover some aspects of cell biology. So I'm not expecting to go over what is a cell. You should understand that a eukaryotic cell, which has a nucleus, which contains DNA, a plasma membrane, and between the plasma membrane and the nucleus is the cytoplasm, and there's a bunch of stuff in there. You're probably familiar with some of it, like mitochondria, the powerhouse of the cell, makes ATP. Well, there's a whole bunch more than that, which I'll talk about in the next lecture. You're probably a little bit familiar about the endoplasmic reticulum. And we'll talk about some other aspects that you may not have covered, like secretory vesicles. We'll talk about lysosomes. You may or may not talk about peroxisomes, but that's okay, because there's enough to talk about with these other organelles. So the expectation is that we do have a basic understanding of what a cell is. So I'm not going to go into that in any great detail. So how do we study cells? So obviously, tissues in our bodies are composed of billions of cells. How do we try to understand how a skin cell functions? Like, we look at it, we try to get a magnifying stereo microscope and see it in a little more detail. It's not going to tell me too much. So we have to have a way of taking cells perhaps outside of the body and then recreating the type of environment that they would be in an organism, and then to study them in more detail. So that's where cell culture comes into play. So this is basically a way to grow cells outside of the tissues from an organism under very strict controlled conditions. And again, we're trying to manage some of the conditions that we would find within the body. And then again, if it's a mammal, mammals maintain about most mammals maintain the body temperature about 37 degrees. So we're going to put them in an incubator. It's going to keep them at a temperature that's pretty close to what you would normally have within tissue. And when we take out cells from a tissue, it's a conglomeration of many millions, trillions of cells. So we need to kind of break them all apart. Most tissues are kind of, the cells are all stuck together. And they're stuck, one of the ways they're stuck together is sort of stuff that's secreted outside of cells called extracellular matrix. And so that helps cells sort of stick together. So we need to break those cell-cell and cell matrix interactions. So the first step we would do is take the tissue and usually would kind of almost like mince it up with a scalpel or a razor blade. And then so you mechanically fragment it. But that just breaks it down into humps. So now we need to sort of break those humps of cells apart. So you would use an enzyme called trypsin. So trypsin is an enzyme that acts like a little lawnmower. It cleans all those proteins away from the outside of the cell, so that those proteins which help facilitate the stickiness of how they stick together are now broken. The other thing we can add is a chelating agent called EDTA. And what does that mean? It soaks up ions, magnesium, calcium. And those ions help the sticky proteins be more sticky. So how do we break up the cells? How do we make the cells come apart? Well, three ways. One is we kind of try to mechanically break them apart. But that only breaks them into clumps. How we break the clumps apart is using the enzyme trypsin, which acts like a lawnmower and cleans all those sticky proteins off from the outside of the cell. And to help with that process and make the sticky proteins less sticky, we add that chelating agent, which acts like a sponge to suck up those ions that help the sticky proteins function and be more sticky. So we simply take away those ions and make the proteins less sticky. So now that we've kind of dissociated those cells, they now need to be grown in a kind of liquid bath, which we call cell culture media. And that media contains many things: amino acids, minerals, vitamins, salts, glucose, which is really important for providing energy to the cells. The carbohydrate is taken up by cells and then broken down. But in order for the glucose to be taken up, it's going to, the cells need a signal to help promote glucose uptake. And in this case, we add what's called serum. So it's a blood-derived product. It would come from, usually we get it from cows, and sometimes it's from fetal cows. It's called fetal bovine serum. And the serum, so that's basically the liquid component of the life. We remove the cellular component, what's left over is serum. It contains all sorts of good stuff, like growth factors. And growth factors can act on the cells, and we'll talk about how growth factors work much later in the course, to help trigger the cell to divide, to make sure it undergoes cell division. And then it also contains insulin. And so insulin is really important to help promote the mobilization of glucose transporters up to the plasma membrane. And that allows glucose to go to the outside, and cell culture media, to inside. And then the cells can break it down and make energy, only ATP. And as I mentioned, we grow these cells. So this is actually an incubator in my lab, and it's set to 37 degrees again. That makes the temperature that these cells can come from, particularly if it's from an animal. And then finally, those tanks there are full of carbon dioxide, CO2. Why would you have that? Well, the CO2 is actually really important for maintaining the pH in the media. You want to maintain the pH in the media kind of neutral with about 7.2 to 7.4. So the CO2 interacts with bicarbonate, which is found in the media itself, and it creates kind of an acid-base equilibrium, which helps stabilize the pH. So it's really important. So how do you know that your pH is good? Well, you can put a pH probe in there, a pH choice, so we recommend it. We have a convenient thing called phenol red. So when we look at these cells growing, and we'll talk about these different culture vessels, that red pinky stuff is not blood. It's actually a dye that's added into the media called phenol red. And it's a pH indicator. So you kind of want it to be kind of pinky, slightly reddish. That means it's a kind of neutral pH. If you open up your incubator and all your media is yellow, that's bad. That means your cells are really overgrown and starting to produce lactic acid because they run out of oxygen, or they've been overtaken by bacteria. And bacteria, again, consumes everything in the media and start to acidify. The opposite is if something's wrong, like your CO2 tank runs out, what can frequently happen is you open up your culture incubator and the media is purple. And that means it's alkaline, the pH is too high. So the types of things that we grow cells in are either dishes, cell culture dishes. These are not petri dishes, they're actually for growing bacteria. We do not grow eukaryotic adherent cells in petri dishes because they won't grow. So the cell culture dishes are either treated with something that helps the cell stick, they use some extracellular matrix proteins, or they use something called plasma treatment. It puts a charge on the bottom of the plates, and that creates the ability of the cells to adhere. So we either have it in these cell culture dishes, so we can take the lid on top, or a flask, where we have an unscrew lid and then we put the media and cells in, and then they float down and then adhere onto the bottom of these culture dishes. But sometimes, and we'll talk about later when you might want that, you don't want cells to adhere. You want cells to grow in suspension. There's various reasons that you might want to do that. When we talk about growing stem cells, sometimes we want them to grow in suspension. Sometimes there are cells that, like blood cells, for example, normally don't adhere to anything in our body. They're continually floating around in our circulatory system. So we may want, in certain conditions, to prevent the cells from, of course, gravity allows them to float down and adhere to the bottom, is that we prevent that from happening. So we use spinner flasks, that's one case. So there's these wands, and they're magnetic, and under here is an instrument that has basically a motor that turns, and has a magnet as well, and through magnetic attraction, it causes these little spinners to turn around and around. And it just slowly happens to agitate the media. And that agitation prevents the cells from appearing. The other way that you can do it is into roller bottles. So you put your culture media cells in a bottle, and you put it right on the side, and then there's rollers, and they basically turn that flask, that roller bottle, around and around, and it's like tumbling your dryer. The tumbling prevents the cells from appearing. Okay, so what are the types of cells that we grow in culture? So as I mentioned, maybe you're interested in some particular organ, or maybe want to culture skin cells, or in this case, this is human aortic smooth muscle cells. So you can take out some of those tissues, and as I talked about, dissect them and trypsinize them, and then put them onto an aeric culture dish, and then allow them to grow in the cell culture ingredient. And what happens is the cells over time will proliferate, and they will fill out the dish. And so what happens is over time, this monolayer, so it's just a single layer of cells growing on that dish, will divide and divide and divide and start to fill up the plate until they become confluent. That means they're all stuck together. And once they reach that state, they no longer continue to grow. It's called contact inhibition. So this is what we call primary cell culture. So they're cells directly taken from the organism, tissue in the organism, and they can be propagated in culture. So when they hit this state where they undergo contact inhibition, you can add a trypsin to the plate, and that will cause the cells to detach and float out and round out. And then you can put them, you can dilute them into fresh media at a low density, and then get them back to this state, and then allow them to grow in culture and fill up the plate. And then you do it all over again. It's called passaging of cells. But the problem is you can only do that passaging so many times. Maybe, depends on the animal, the tissue. It can be anywhere from 10 times to maybe 50 times. And at this point, they'll undergo contact inhibition, and then you'll dilute them and plate them onto a fresh plate, and they don't grow anymore. They reach what's called the Hayflick limit. And this is in part related to something called telomeres. How many people here are familiar with telomeres? I think you should have covered it first for biology. So those are the ends of chromosomes, and every time a cell divides, those telomeres, those ends of the chromosomes, get a little shorter, and they reach kind of a critical state where they're so short it actually causes this this phenomenon called Hayflick limit or cellular senescence. They simply can't divide any further. So that's one of the limits when you use primary cell culture, is you can only grow it for so long in culture until you reach the Hayflick limit. So it's kind of a laborious thing making primary cell cultures. You have to do it all the time. You can do a certain level of experimentation, maybe for a month, and then throw it all out, and then go get fresh tissue and do it all over again. It's kind of a pain in the butt. So what would be great is a cell line that can grow indefinitely in culture. Cells that can grow indefinitely in culture. And that leads us to cell lines. So these are transformed. What does that mean? They've undergone some kind of genetic transformation or mutation, which overrides a normal cell cycle regulatory control, and also the mechanisms that elicit Hayflick limit. So I won't get into it because there's a number of complicated ways that cells can escape from this Hayflick limit, but cancer cells quite frequently do that, and that's what allows them to grow uncontrollably. They can grow uncontrollably in their body, or, as we'll talk about with Henrietta Lacks, and a fellow by the name of George Guy, took tumor cells and managed to propagate them in culture. So this leads us to an interesting but somewhat tragic story about a cell line called HeLa. And the cells are actually named after a woman called Henrietta Lacks. So this was a poor woman living in Baltimore region in the 1940s and 50s. In 1951, after giving birth to a number of children, she was having a lot of abdominal pain, and went to Johns Hopkins Hospital, one of the few hospitals that would treat Black patients then. And upon examination, it was determined that she had a very aggressive, pronounced cervical cancer. And so the physician took a biopsy of that cervical cancer, and he gave it to this researcher, George Guy. And George Guy had been trying for many years to try to create this exact thing, cell line. He'd grown many different types of tissue cells in culture, but would never get them to grow indefinitely. And he had tried some other cancers from patients, and for whatever reason, they couldn't grow in culture. And remember, it was kind of a guessing game about how to grow cells in culture, even in the 1940s and 50s. They were still kind of playing around, trying to find the magic ingredients to get cells to grow. So they didn't quite have it optimized just yet. But when he got a hold of Henrietta Lacks's cancer cells, they did something that never been seen before. They grew, and they grew phenomenally well. And this was incredibly unusual. And he could propagate them in culture indefinitely. They were, in essence, immortal. They could grow indefinitely. And so this was a major development in cell biology research, because now we had a tool that we could grow very easily in culture. It was very reproducible. The cells that are growth, they could divide really quickly, and it didn't take a lot of special media to grow them. So he was very enthusiastic about it, and he did give it out to many labs worldwide whose researchers were desperate for something like this. And so these HeLa cells went on to make major discoveries in biology, basic science, and also medicine. So they were used to help understand basic genetic principles, to understand how cell growth occurs, understand cancer biology. They were used to screen for anti-cancer compounds. They were used to propagate viruses. They went into outer space to understand how zero gravity affects cells. So they made phenomenal discoveries. In fact, some of those Nobel Prize discoveries in part have some lineage tracing back to HeLa cells. What's really unfortunate is that Henrietta Lacks died at the end of 1951 and was buried in an unmarked grave. And the family was quite poor. And what's really quite tragic is that these HeLa cells were also licensed out to pharmaceutical companies that used them for drug development that made a lot of money for these pharmaceutical companies. And none of this ever was translated back to Henrietta Lacks or her family. In fact, those cells were taken without her permission. Nobody knew. Henrietta Lacks didn't know, her family didn't know. Her family discovered many years later what happened is the HeLa cells grew so well in culture that they started contaminating over time, other cell lines were developed. HeLa cells were basically another cell line. Once it got out, you could do this with certain aggressive tumors. Other people started making cell lines. And they were growing HeLa cells and some other cancer cell line. And what was happening is the HeLa cells were, in fact, the HeLa cells grow so well, they were dust that can occur in a lab and even in the cell culture incubator. The HeLa cells can actually attach to the dust and float over and contaminate other cultures. And so this was a big problem, is that they had to figure out, well, what is the original HeLa cells versus what is contaminated cells? And so they contacted her family, her children, surviving children, and asked, can we get some of your, can we look at, get some cells from you to compare genetically to your mother, what we were talking about? And they also contacted her husband, who had a very rudimentary education. And his understanding was that your wife is still alive and she's living in a cell. And so there's a whole bunch of ethical problems in terms of consent. You don't take tissues from people without their consent. You do not contact family members without taking into consideration how this would impact them. And so what this, so Henrietta Lacks, probably her name would have disappeared if it weren't for a major part, not the only person. An author by the name of Rebecca Skloot, in 2010 published this book called The Immortal Life of Henrietta Lacks, which is a New York Times bestseller for like a couple of years. And so she kind of did an expose on this, and this really got a lot of interest back. She contacted the family members and actually kind of helped as an advocate for them to seek compensation. And as of 2023, they finally got some compensation over 7 years after Henrietta Lacks' cells were taken from her without consent. Thermo Fisher, which is a major science, big scientific company, has settled with the family. So there has been some compensation. Anyways, I would recommend that you read this book. It's really quite an impressive story. And Henrietta Lacks' name is not like it. It's now, her name is now on various elementary schools and other institutes. So she did get her name on that. Okay, so we talked about primary cells. So one of the, again, it comes down to like, what are you interested in? And if we're looking at what is called normal, non-diseased, primary cells would be a good cell type to look at. So they also morphologically are quite different from transform cells. Transform cells like amyloid cells that have mutations, grow uncontrollably. So if we were to, for example, take fibroblasts, these, in this case, I believe there's mouse fibroblasts, skin cells. And you grow them in a dish that has a very flat morphology, they grow sort of in parallel arrays, they're lined together, and they exhibit contact inhibition when they get really close. You can take those same cells, primary cells, and introduce an oncogene. So using a virus, you can express an oncogene. And we'll talk about that towards the end of this course. But basically, these are genes that, when activated in cells, cause uncontrollable cell growth, i.e. makes them transformed. We're mimicking what happens maybe under normal circumstances with random mutations. We introduce a mutated type of gene that regulates cell cycle into the cell. They now become transformed. Their morphology looks quite a bit different. In this case, they're more rounded, not so flattened. Then we have immunity projections. They tend to grow all on top of one another. And I've seen cancer cells in the dish where they form actual clumps so you can see with the naked eye. You can grow back. And that's when they grow on top of one another. And then they lose contact inhibition. So once they can get really close together, it just doesn't stop them from growing. The only way to stop them from growing is if there's no media or nutrients available anymore. So those are two sort of morphological comparisons. But it's also important to know, it depends on what you want to study. If you just want a cell line that grows quickly in culture, maybe you're interested in the metabolic process, using a transform cell line is more convenient. But you have to keep in mind that it's cancer. If you want to understand cells that are non-cancerous cells, you may have to go on to primary cells. So as a cell biology researcher, we frequently use both, depending on what you're interested in examining. So the other thing to think about is when cells divide is just like a HeLa cell. It divides and it gives rise to two identical HeLa cells. So that's an example of symmetric cell division. So it could be primary cells, like skin fibroblasts, that's not transformed. And it will also undergo symmetric division and give rise to more or less two identical fibroblasts. So that's kind of more typical of what we see in most cell culture types of situations. But you can also get a situation where cells can divide asymmetrically. What does that mean? Well, it simply means that a cell can divide and it can go under one of two circumstances. It can give rise to two cells, but the two cells are completely different phenotypically from the originating cell. So it can give rise to two cells that have different properties from the originating, I'm going to call it the mother cell. So maybe this is the mother cell and two daughters. The mother gives rise to two daughters, look totally different from one another and different from the mom. Or you can have a cell that gives rise to two cells, one cell that's identical to the originating cell and one that's different. And so where you tend to
question. So more like cells and culture, are you talking more like cancer cells or the one that divide?
Okay, so mortalized cells in culture, right? So that could either be, how do you have mortalized cells in culture? Two different ways. One, they can come directly from a biopsy of a tumor, like Henrietta Lacks, or we can use genetic tools to artificially overexpress an oncogene that overrides how that cell normally divides and grows, that makes it become transformed and immortal.
Doesn't that maybe a pancreas cell lack control and there's more mutations, so the two are not identical?
Well, you're kind of getting into a different branch. Okay, yes, they can accrue more mutations over time, and they can adapt a different phenotype, but that's usually in culture over many, many passages. I'm getting into genetics. I'm going to stay with that. So I think more just to understand the basic principle. So symmetric division, transformed cells, primary cells, they divide, they give rise to two identical cells. That's fine for experimental purposes, doesn't matter. When it comes to something called stem cells, which I'll talk about in a little more detail in the next few slides, stem cells can give rise to different types of cells. And we're going to talk about the different flavors of stem cells. And so when we look at a stem cell, it can give rise to maybe two identical stem cells. Stem cells can do both symmetric cell division and asymmetric cell division. And I will give examples of when it does both of these types in the next few slides. So just bear with me. So a stem cell can give rise to a stem cell, and then it can give rise to a cell that's kind of veering away from being a stem cell. It could be what we call a progenitor cell. And I know this causes great consternation among students. What's a stem cell? What's a progenitor cell? Well, a progenitor cell is a cell that starts to become more committed to being a different cell type, becoming different from the stem cell itself. But the progenitor cell has not gone all the way to what we call the final state, which is a differentiated cell. That's a cell that's it. It's reached its final destination. It's not going anywhere else. If it's a liver cell, it's a hepatocyte. That's a liver cell. That's all it does. It's a one-trick pony, and it does its function. If it's a muscle cell, it's a differentiated muscle cell that can involve in contraction processes. But those cells, those differentiated cells, don't just pop out of nowhere. They have to come from somewhere. So they typically come from stem cells, but there could be some intermediate step where they exist. These stem cells, they're not stem cells and they're not differentiated cells, they're progenitor cells. They're in the intermediate stage. And the progenitor cells themselves can give rise to different cell types. They can go asymmetric or symmetric division. It'll become a little bit more clear when we talk about adult stem cells in a minute. So this process of dividing into both symmetric and asymmetric manner is important for a number of processes, either embryonic development or in adult stem cells, which we'll talk about in a minute. So particularly in embryonic cells, during an early embryonic development, cells have to, sometimes they need to amplify the stem cell population to make a big enough pool that then can start to differentiate into different structures that will go into forming bone or muscle or intestine. And so there's a lot of intermediate stages that can happen, and that's where the progenitor cells come into play. I don't think I know students get really hung up on this progenitor thing and find it very difficult. I don't think it's really critical to focus so much on the progenitor, but understanding that stem cells are important for giving rise to different tissue types, or in the adult cell stage, they give rise to certain specialized types, which I'll talk about in a minute. And there's some intermediate stages where you have not really stem cells, but not differentiated cells, intermediate. Okay. So let's get on to embryonic stem cells. Where do they come from? The name kind of tells you, they come from embryos. So when an egg and sperm fertilize, they form a zygote, and that starts to form an early stage embryo, and then it forms this complex structure called a blastocyst. And there's a collection of cells that form on one side of the blastocyst called the inner cell mass. And if we were to go in with a pipette, using a microscope, scientific pipette, you can pull out those inner cell mass cells, that clump of cells that develop blastocyst. You take it out, and then you put it into culture. And the dish that you put it on has already been preceded with fibroblasts. So these fibroblasts, they're called fibroblast feeder cells. And why do we need to put them on fibroblasts? Because the fibroblasts are secreting all sorts of beneficial factors, survival factors, and growth factors that allow the embryonic stem cells that are found within the inner cell mass to adhere to those fibroblasts, and then also to survive, and then also to grow and divide. And so quite a few of these inner cell mass cells will die. They don't take well to culture, but the ones that do, you can then pick that colony, or that clump of embryonic stem cells, use trypsin to break them apart into individual cells, and then put them back onto a new feeder cell plate. So the individual cells fall down onto the plate, they adhere onto the fibroblast feeder layer, and then they start to grow. And so the ones that grow, you can pick them and put them again into another plate, the fibroblast feeder cells. And then now they can, in essence, become an embryonic stem cell line. Now, in theory, they can grow indefinitely. Does that mean they're transformed? No, they don't have mutations per se, but that's part of embryonic stem cells, that they have an ability to proliferate. Okay, so we're taking it from an embryo, will it ever turn into a mouse? The answer is no, because it needs these other cells, the trophoblast, and there's other cells in there to actually form a full developing embryo. So what it is it's going to form embryo-like cells, but they can never form a full embryo under normal just growth conditions. And yes, they are kind of like, are they triplets? I don't know. They're very similar, probably genetically very identical, very similar to one another. In culture, you can get some genetic drift, but let's just assume that they're basically embryonic-like cells growing in a dish, and you can continually passage them as long as you maintain them religiously. Very difficult to grow these for a long period of time without them starting to spontaneously differentiate. So in theory, they are immortal, but in practice, they're very difficult to passage like 100, 200 times like human cells, because they start to differentiate, because it's so difficult to maintain them under these controlled conditions. That's more of a technical concern. I think it's more like, who cares? What are you going to do with these cells? You can do a lot of things with these cells. So if we don't want to understand aspects of embryonic development and a cultured environment, that's what we can use these cells for. So we can take these cells and we, instead of growing them on adherent fibrous feeder layers, we grow them in suspension. And when we grow them in suspension, they form these big balls called embryoid bodies. And it actually kind of starts to force them back to thinking about to being an embryo. And when you take those embryoid bodies, you can then grow them under different culture conditions with certain growth factors and signaling molecules to direct them towards certain types of tissues. This is what we call the three germ layers, the endoderm, the mesoderm, and the ectoderm. And so this could be a variety of gut cells, cartilage, neuronal cells. So we can maybe understand some embryonic developmental processes by using these embryonic stem cells, grow them as embryos, kind of trick them back to thinking they're like an embryo. They're not really sure. And then put them in defined culture conditions with certain growth factors and signaling molecules to force them to go into certain germ cell layer type cells. And so this, again, is from a sort of basic science, has a great advantage to understanding basic mechanisms of how, what we call cell fate. How does a cell know to go towards being a bone cell, or a neuron, or a muscle cell? And identifying the factors that actually control those processes. Embryonic stem cells play a major role in figuring out what makes cells know where to go. Now, the other thing is that we can grow cells as a adherent monolayer. So it's a layer of cells growing flat in the dish. But cells don't grow like that in our body. So now more people are becoming interested in trying to mimic some of the processes that happen in our body normally, or how as tissues form. And so this is where it's led to 3D culture. So you could take those embryonic stem cells and you could grow them in suspension culture that would form those embryo bodies. And then you could take those embryo bodies and actually put them in, it's like putting a golf ball in a big bowl of jello. Right? So you plop it in there and it kind of sits, floating around, not floating, but kind of quasi-floating, stuck in the beaten gel. So you could take extracellular matrix type of matrix. You can create kind of a gelatinous beauty that's got extracellular matrix proteins insert those embryo bodies in there, and then it will continue to grow. And depending on the type of growth factors and signaling molecules you put in the culture, can now start to form organoid tissues. So these will be called organoids, which would maybe start to more mimic the types of tissues we find in our body. So here's an example of a brain organoid. So these were stem cells. So one thing that I should mention, it's really important to know, going back here, is embryonic stem cells are pluripotent. What does that mean? It means they can give rise to the 3 germ cell layers, which can give rise to basically every tissue in the body. So pluripotent stem cells really cool cells that can basically give rise to any cell type in your body. And so if you take those pluripotent stem cells and you can put them in 3D culture and give them the appropriate types of growth factors and signaling molecules, they can start to form organ-like structures. In this case, they can start to form a brain-like structure. And so it will progress in a manner that's very similar to what we would see in a fetus. But it can't go, you can't basically have a functional brain thinking in a dish. It doesn't work that way. It will grow and form structures that are very similar to an embryonic or fetal brain. And then we'll start to differentiate into structures that we actually find in the brain. And so, again, this is very valuable for understanding developmental processes, but also generating experimental models that are much more similar to the types of organs and tissues in the body. Because I can tell you, the brain doesn't grow like, our brains aren't like cells in a dish. They grow, it's the three-dimensional structure that has very complex structures, and different parts of the brain have different neurons and different cell types. And if we were, for example, wanting to test a drug to treat a brain disease, we quite frequently do that in adherent monoculture with neuronal cells. But very rarely does that actually translate into a drug that works the same way on the brain. So we can test drugs in these brain organoids, and it's more likely to elicit effects that would likely happen in an actual brain, in the brain itself. Now, Dr. Pemmican will probably do a little bit more lecture talking about what's all this fluorescent stuff. There's ways that we can label certain cells or certain structures inside the cells using fluorescence microscopy, which we'll talk about in more detail. But basically, we can label, for example, stem cells are indicated in red, and differentiated neurons are in green. As you can see, these inner parts of this organoid is enriched with neural stem cells, and it gives rise to neurons which move outwards and accumulate at the periphery. So again, which is kind of similar to what we see in fetal brain development. Okay, so the other, we talked quite a bit about embryonic stem cells, which are pluripotent stem cells. Another important cell type is adult stem cells. So adults here will have stem cells in their body in various tissues. And these stem cells play a really important role in maintaining our tissue structure. So I'm going to use the intestine as one example. In this case, when we look at a section of our intestine, and we look at, so the intestine is a long tube, imagine cutting the tube's running this way, we cut it this way, and then we look at the line, the outer part of the tube, it's composed of intestinal epithelial cells, and they kind of go in this undulating pattern. So these little finger processes are what we call villi, and so they're all lined with cells, intestinal epithelial cells, except at the bottom, where my fingers are, at the bottom of the villi, are called crypts, intestinal crypts. And that's where we find these intestinal stem cells. Now, the textbooks has some other stuff in there, which I'm not going to talk about. It's just important to know that these are adult stem cells found within the crypt. And they can get signals to either self-renew and divide symmetrically and create more stem cells, or you may get a signal to divide asymmetrically and give rise to a cell that could be like a progenitor cell. It's not a stem cell anymore, but it's not a fully differentiated epithelial cell. So these progenitor cells could then undergo symmetric division and divide a number of times. And then as they divide, they sort of push, the cells start to push and work their way up, to be like the fingers, so to speak, until they become a differentiated intestinal epithelial cell. So this amplification or this proliferation of these progenitor type cells helps push the cells before them upwards. They function up here at the tips of the villi to help absorb nutrients and food from the lumen of the intestine. And then at the end, what happens is they die off, usually by a process called apoptosis, which I'll program cell death, which I'll talk about much later in this course. And they basically get shlucked off and basically pooped out, keeping up a lot of intestinal epithelial cells. You know that, right? So what's important to know here, and again, this happens quite rapidly. So your intestine is actually a very active area of proliferation, cellular proliferation. So much so that you replace your intestinal epithelium about every 5 days. So it's important to maintain your stem cell population at the bottom, so those adult stem cells need to self-renew, divide symmetrically a little bit to maintain the pool of stem cells. But they also need to divide asymmetrically and give rise to progenitor cells, which will probably most likely divide symmetrically for a little bit. And then those progenitor cells now to turn into differentiated, mature intestinal epithelial cells, which are important for absorbing nutrients from the lumen of the intestine. So that kind of is an example of asymmetric and symmetric division and why it's important to regulate. If, for example, we had stem cells here that only divided, that maybe divided and only gave rise to progenitor cells or differentiated cells, and didn't self-renew, you would deplete the stem cells. And that would cause a lot of problems in your intestinal epithelium, because you always need that reserve of stem cells to help replenish the worn, washed out cells at the tips. Okay, I'm going to just introduce this concept. When we think about stem cells, we think it's basically a one-way, particularly pluripotent stem cells. We think it's a one-way ticket. If you're on a mountain and you want to ski down, so you're the stem cell that's there, and you give rise to other cells that they differentiate, and they only go one way, and that's downhill, or it's differentiation. Maybe at the top you hang out there and you have a cup of cocoa, and you give rise to another stem cell. It's called self-renewal, which we just talked about. But once you initiate the step of differentiation, the idea has been that once cells are differentiated, they can't go back. And we now know, and I'll just mention it here in a minute, that that's not true. We have a way of taking differentiated cells and reversing them back to a pluripotent stem state, which is called induced pluripotent stem cells. And in the next lecture, I'll talk about some of the genetic tricks that we use to help revert differentiated cells back to pluripotents. I'll finish there.